I think we need to know what is the normal hearing process. So I'm going to spend the first few minutes and the next 4 or 5 slides in explaining to you how do we hear. Now this is an exquisitely beautiful mechanism. I'm very fascinated by that. Around 200 genes are involved in the process, and this is one process in which very clearly the mechanical energy is converted into chemical energy and then into electrical energy, and then into perception of what we hear. The ear, which is the main organ of hearing, okay. So the main organ can be divided into the external ear, the middle ear, and the inner ear. The external ear actually captures the sound waves as they come in, and then it goes into the inner part of the ear, which is called cochlea, and this is actually the sound box. The cochlea is the main part of the ear which converts mechanical energy into chemical energy and then finally into electrical energy. This is this has 3 semicircular canals and 2 of the smallest bones. The cochlea here is actually the sound box in which the conversion takes place, and if you make a cross section of that, you see this kind of a picture with scala vestibuli at the top and scala media. Scala media actually shows you potassium ions here, and it is the increase in the potassium ion which converts chemical and then electrical potential. I'm going to show you a movie in a minute, but this is just to okay, so now one second. So as you can see here, the sound waves you see, they are coming in, and the spinner actually captures them and then passes on to them. And these are the 3 smallest bones in the body, and you can see just like a house bell, you see it is falling on the tympanic membrane here and vibrating it, and then this is causing waves which will then fall onto the organ of Corti, which is the sound box, like a sound box in a tape recorder. And you can see here, you see that this blue is being converted into red, and that actually shows you the difference in the ionic potential, which will be shown more elaborately in the next one. What happens is, you see, that the here in the cell, they are, with the impact of the sound waves, you see, they are shifted, they are migrated, and at the back, there is a plate there, template plate, and this, as the plate, you see, as it lifts, it allows the potassium ions to pass through, and this transfer of potassium ions actually creates the potential difference. You see? Now, these are 3 rows of hair, and as they vibrate at the bottom here, the potassium ions, they move into the now this, if you see watch closely, you see, I mean, this potassium ion build up, and then you see the electrical signal here. This electrical signal goes through the afferent nerve to the brain, and then the brain receives the perception. This is actually the summary of the whole movie here. The stereocilia are vibrating, and as they vibrate, you can see here, potassium ion is forced into, and this potassium ion creates a chemical potential here, plus potential, which is then converted into electrical and goes to the brain. Okay, now what is hearing loss? Normally, a normal person, first of all, the hearing is measured in decibels, and 0 to 25 decibel is the normal hearing. And then as it goes on, you can see profound hearing loss is about 100 decibel, and any sound above 125 decibel is extremely loud, and it is painful, and you cannot hear that. So I'll be working in the range of 0 to 100 decibels. There are two kinds of hearing impairments. The first one is conductive, and this is generally in the outer or middle ear. Sensory neural, and again most of my work is on sensory neural, and this is confined to the cochlea or the inner ear. You can also have mixed hearing loss, but the results that I'll be presenting you relate predominantly, if not exclusively, to sensory neural. Now, deafness can be caused genetically or environmentally. 50% is generally by environmentally, and the remaining 50% is genetic. Of this 50%, about 30% is syndrome. So that means you see that in addition to deafness, you have other syndromes, like Pendred syndrome or Barber syndrome. But my presentation will be exclusively on non-syndromic deafness, which is about 70% of this 50%, which is genetic based, and this is non-syndromic deafness. Non-syndromic deafness means you see that the person only has a hearing impairment and absolutely no other syndrome. Now, when I started, the autosomal recessive loci, they are given numbers like DFNB1, DFNB2, DFNB3. And when I started in the year 2000, the first locus that we discovered that was designated as DFNB26. And last month, we registered another one at DFNB94. So during the 10-year period, about 70 loci have been added, and I will show you the contribution that we have made in this 70 loci. Now, the main goal of my work has been to find out which proteins are involved in the hearing process. I said in the beginning that there are about 200 genes. So you can imagine, you see that there are at least 200 proteins which are involved in the hearing process, and a single base mutation in any one of these 200 genes can cause deafness. And when it causes deafness, then the person don't speak. There is absolutely no syndrome, and this is not an impairment, but simply the fact that you cannot hear, you see, you cannot speak. We have studied this by forward genetics. You mentioned this morning. We use linkage analysis to map the disease gene to a particular chromosome, and through position cloning we identify gene mutations that segregate, and then find out, you see, which protein is involved, which are the partners, which are the other proteins which interact, and finally, how do they help or how they contribute in the hearing process. This is just a cartoon showing the same thing here, you see. But the important thing that I want to show here is that we are looking at the protein partners, how do they make the complex, which actually results in the hearing process. Now, the work plan, identification of families, which is the main task, and we have enrolled close to about 2,000 families that we have worked in the last 10 years. We do the linkage analysis, we call that as exclusion studies. analysis, linkage to the known genes, and then genome-wide scan. And then once we have identified the region or locus, then we try to narrow it down, do the positional cloning, find out the mutation, and then do the animal studies, which is cell biology, knockout mice or other animals. Our major strength is availability of large number of consanguineous families. Now, Pakistan is, has a very unique population, and as we know, you see, it used to be part of the eastern part of India, and the Indian subcontinent